Welcome everyone to the NPTEL course on Food Science and Technology. So this course is instructed by Professor H.N. Mishra sir, who is a professor in the Department of Agriculture and Kharagpur, and I am Shikha Sharma, a research scholar in the Department of Chemical Engineering, IIT Guwahati. So this this live interaction session is basically for week 7. So the assignment deadline for week 7 is 11th. So kindly submit your assignment on time. Uh, the topics that we will be discussing in today's session or today's class is basically the module 7, which includes basically microorganisms and the associated like their how these microorganism grows and what are with the food, both in terms of the positive effects and the negative effects. So it includes basically introduction to organisms, basically bacteria, yeast, and molds, then microbial growth, kinetic model, factors influencing the growth of the microorganisms in the food, such as water, pH, and other factors, and then microbial death kinetics and factors affecting, and after that, about the probiotics and bacteriocins, and then microbial spoilage of the foods, and last is the food poisoning. So it includes basically lecture 31 to lecture 35, which includes lecture 31 includes about food microorganisms, and 32 about microbial growth, 33 about microbial spoilage of the foods, and lecture 34 about prevention of the food poisoning and the spoilage, and lecture 35 about beneficial. So in this interaction session or in this tutorial lecture, we will be basically solving some of the numerical problems, also about the d values, z value, and rest will be the theoretical questions. So if you have any doubt, please feel free to ask. Uh, so let's begin with the today's first question. So which of the following is a characteristics of gram-negative bacteria? So we have four options about thick peptidoglycan layer, B retains crystal violet dye, C contains lipopolysaccharides in the cell wall, D during reproduction. So it basically microorganisms in the food. So there can be probiotics, prokaryotics, and second can be eukaryotics. So the prokaryotics one, they are the one which are single-celled microorganisms, and they are having the not properly defined or lack of the defined of proper nuclei or other organelles. So it includes basically the bacteria. It can be of any shape like spheres. So that can be cocci. Rod shape includes bacillus, and spiral shape includes spirochetes. So eukaryotics basically they are also the single-celled organisms like the yeasts, and molds also includes in this category, which are multicellular organisms, and they have the nucleus, and they also have the other organelles or organelles. So it includes basically the yeast and molds. So these type of the microorganisms can be associated with the food. Now we're talking about here the bacteria was in the first question, that which there is a classification of the bacteria. The bacteria can be gram positive and it can be gram negative. So the characteristics of gram negative bacteria, we need to tell which among these four is the characteristics of the gram negative bacteria. So anyone who would like to answer this question? Anyone who would like to answer? What are gram negative bacteria basically? Okay, let's see. So basically, bacteria contains lipopolysaccharides in the cell wall basically. So bacteria, as we discussed, they can be gram positive and also gram negative. So basically what happened is there is this test which is gram staining. So gram staining test, it is introduced by basically Danish bacteriologist Hans Christian Gram in 1884. So in this test, this gram stain test, what happens is first the bacteria will be put on this glass slide and it is air dried. So smear kind of like the bacterial smear can be prepared on the glass slide. So this bacteria is basically there after that will be stained with this primary stain which is crystal violet, and after that it will be washed with the excess crystal violet will be washed with that water, and then another agent which is the fixing agent or the uh, those like this is the fixing agent or we called as moderant, or in this place we can use iodine solution. So after that iodine solution will be put over that, and after that the decolorized for decolorization or decolorization agent we use basically 95% alcohol or acetone. After that the at the end counter stain will be done using the safranin. So what happens basically in the gram positive bacteria and what happen in gram negative bacteria basically. So gram positive bacteria, these kind of bacteria, they have the thick peptidoglycan layer, so they retain the crystal violet dye, and that's why they will have the violet or sorry the purplish or the violet color, because they can have this thick peptidoglycan layer, and they can because of the layer they can basically retain the primary stain which is the crystal violet stain. And this type of the bacteria, the gram positive bacteria, they have also the ability to form spores, whereas on the other hand the gram negative bacteria, they will have the thin peptidoglycan layer. So because of that it will not be able to hold the primary stain when we are using the decolorizing agent. So because of the decolorizing agent, that primary stain, which is the crystal violet, it will be washed off, and the second stain, which is the counter stain, safranin will be this bacteria. So gram negative bacteria, they have the glycan layer, which is complemented lipopolysaccharides layer in the it has peptidoglycan layer plus it has lipopolysaccharides layer in the cell wall. So due to the absence of the lipopolysaccharide layers, which restricts the absorbance of the crystal violet dye, giving the negative reading for the gram staining. So because the crystal violet color, which is the primary stain, will not be able to retain by these kind of bacteria. So they lose the crystal violet color during the decolorization process and takes up the safranin color during the counter staining or the second staining process. So if we see the process, basically what happens when we are putting on the glass slide the bacteria like the gram positive or the gram negative. So when we are using the crystal violet prime, after that we are using iodine solution, and then the ethyl alcohol, or we can use ethanol, or we can use acetone, and at the end we are using safranin. So gram positive bacteria, they will be able to retain the purple color, whereas the gram positive bacteria, they will be able to retain the pinkish kind of color. So here is the image on the microscope. If we see that it is a gram positive and this one is the gram negative. So we can see here in the gram positive bacteria, this is the peptidoglycan layer, which is this long, thick. Whereas if we see the peptidoglycan layer in the gram negative bacteria, it is thin like this. And this extra, basically this is the thin peptidoglycan layer, which is there in this gram-negative bacteria, and this is the lipopolysaccharide layer, which is complemented with the peptidoglycan layer in the gram-negative bacteria. So because of this, it shows kind of the pink color in the microscope. So here the answer is a thick peptidoglycan layer, which is wrong because in gram negative it is thin and it cannot retain the crystal violet color. But it contains the polysaccharide layers in the cell wall, which is true. And these spores, they are basically produced by the gram positive bacteria, not the gram negative bacteria, because of the thick peptidoglycan layer. So answer C is correct in this case. So gram-negative bacteria, they can be further classified based on their structures, diplococci or cocobacillus, bacillus, coma-shaped. So they can be further classified as then aerobic or non-aerobic, or if it's like it can which condition or which pH range it is producing and which temperature it is producing. Based on that, it can be further classified. So gram-negative bacteria can be diplococci, uh, cocci, or that means there are two coccus in the structure. So it can be aerobic or non-aerobic or anaerobic. So if it's aerobic, it cannot, like if it's aerobic, so it cannot utilize maltose. So anaerobic bacteria, they can utilize maltose. Utilization will be there, so they can be put into another classification. And bacillus, they can basically then cocci bacillus there, which has influenza and other kind of bacteria. Bacillus have the lactose fermenting capability. So if they can ferment the lactose, then they can be classified into another class, this class. And if it will not be able to ferment the lactose, or then if it oxidize the lactose, it can be, and then also if it's producing H2S, then it can be Salmonella, otherwise it can be Shigella. So like that, if it oxidize, um, like there is oxidase, so it oxidize, but it doesn't produce H2S, so they can be classified into the Pseudomonas. Likewise, there is this coma shaped, if it's oxidase positive and if it 42 degrees Celsius, it can be classified as Cigigene, and it can grow in alkali media. It can be a cholera, and if it produce urease, then pylori. So these kind of further classifications are there in the gram-negative bacteria. This is the classification of the gram-positive bacteria. So gram-positive can basically be classified as based on the shape, like bacilli, which is of like rod shape, cocci, which is of round shape, and branching filaments, which has some branches. So branching filament can be classified as aerobic or anaerobic. So based on that, it can be classified based on if they are able to grow in the oxygen or not. Then in bacilli also, it can be aerobic, anaerobic, Listeria, and aerobic is Listeria, like that. And cocci shaped aerobic is it's there. Then if it's catalase positive, it can come under Staphylococcus. And if it's catalase negative, it can come under Streptococcus. So which is further classified as alpha, beta, and gamma, and there is further classification of that. So that means that in the gram positive and gram negative stains, they can also be further classified into different kind of like based on their shapes, and if they are which kind of or which environmental factors they take to grow. So based on that, further classifications are done. Okay, let's move to this second question. Match the component of a microbial cell which is in the column 1. So we have flagella, pili, ribosomes and endospores. They are the microbial cells components, and in column 2 we have the functions like genetic material transfer, protein synthesis or survival during unfavorable conditions. So we need to match here. Anyone who would like to answer this question? Lakshmi B, also Anjali is saying B. So they both are saying B. Let's see. So they both are correct. So basically, bacterial cell structure, if we see, we have flagella or flagellum, if we call in singular form. So it is this structure called the flagella. Then pili or pillars is these kind of structure like this, which is all around the bacteria surface. Then ribosomes, if we see ribosomes, which is like this inside the cell. These are the inside cell components. And if we see uh, endospores, so endospores will be inside the bacteria cell. So it will be inside spore mother cell. So if we see endospore will be having different other like cell components, like it has one core or one, and also it will have in that core it will have some DNA material for the replication, and then it has a core cell wall, and it will have cortex, and after that there is a spore coat which is very hard, and then exosporium will be there. So this is the endospore structure. So flagella basically ensures in the mobility to explore and adapt to the environments. They are long hair-like filamentous structures. So they are basically long and hair-like filamentous structure, so that bacteria can go anywhere and stick to that new surface. In search of food, it can go, this bacteria. So pili is basically short, hollow, non-helical filamentous structure. They are exclusively found in the gram-negative bacteria only they found, and they aid in adherence to the host cell. So it will help the bacteria to attach with the host cell, and they contribute also to the genetic diversity and adaptability via horizontal gene transfer. So it can help in the transfer of the genetic material also. Ribosomes, they are basically the machine life-sustaining protein synthesis, or they can also produce some kind of the RNAs, DNAs, also the enzymes. So bacterial ribosomes are of 70S type and quite smaller than the eukaryotics, which are 80S types. Then endospores, they are produced because of a survival strategy when bacteria faces environmental stress, or it faces tough, so it can go under the endospore formation. So this endospore is a dormant, tough and non-reproductive structure which is produced by certain bacteria from the Firmicute phylum. So endospore formation is usually triggered by lack of nutrients. If there is lack of nutrients, so bacteria will undergo the, or some kind of stress is there, so bacteria may undergo the formation of the endospore. And usually it is formed by the gram-positive bacteria. In endospore formation, basically the bacterium divides within its cell wall. One side then engulfs the other. So basically it will what will going to happen when endospore will form. This is the bacterial cell. So it will go near the cell wall and it will take some cell wall and start formation the spore, and then slowly it will form all different kind of structure which is having proper spore coat with there will be there to protect the bacteria from the tough situations. The endospore basically it will also have the bacterium's DNA part of its cytoplasm, and it will be surrounded with using very tough outer coating. So the endospore can survive in the tough environment like without nutrition, and they can resist the ultraviolet radiation also, and desiccation, high temperature, extreme freezing, and also the chemical disinfectants. So these are this endospore kind of structure is very tough kind of structure, which is like this. So if we see flagella, its function is for the mobility, and pili, its function is for the genetic material transfer. Then ribosomes is basically used for the protein synthesis or the enzyme synthesis, and endospores are for the survival during unfavorable conditions. So B option is correct here. Let's discuss this question. Which of the following statement is correct with respect to the microbial growth curve? So we have about lag phase, exponential phase, stationary phase, and the death phase in the microbial growth curve. So we need to tell which statements are correct regarding that. In the lag phase, cells increases in size but not in number. Exponential phase shows a constant doubling time. Stationary phase occurs due to nutrition depletion, and death phase involves immediate lysis of all cells. Anyone who would like to answer this question? Anjali, and she's saying D is the correct answer, but all the four statements are correct. Lakshmi is saying statement 1, 2, and 3 are correct, but fourth statement is not correct. Anyone else who is giving an answer to this question? Okay, let's see the answer. So C is the correct answer. Let's understand why fourth is the incorrect statement. So microbial growth curve, it basically shows the growth of the organisms reproducing by binary fission. So binary fission is, suppose this is a bacterial cell. So inside this, there will be the binary, like the replication of the DNA or the nucleotide or the chromosome will be there, and after that two copies will form, one copy and two copy, and after that they will uh, come actions like this, and here there will be this fission will be there, and two daughter cells will produce from the one mother cell. So this is binary fission. So microbial growth curve, it basically depicts the growth of the microorganisms which are reproducing using the binary fission, and it can be plotted as the logarithm of the number of viable cells versus the incubation time. So it is the basically the curve, incubation time, how much time is taking to what are the number of viable cells in the, this is basically a logarithm kind of graph of that. So it is performed basically in the batch culture. Batch culture means that we are in the like when we are seeing this how much in how much time, how much is the bacteria producing or the viable cells are there. So we basically consider a batch culture kind of system where the nutrients we give on only one time. We'll give, like this is a fermenter, suppose. So we'll give the nutrients one time, and then we'll let the microorganisms to grow. And in the continuous culture, the nutrient will be given on the continuous basis. But here we'll give just on in the initial phase or the starting phase. So where nutrients will going to decrease with the time because bacteria are eating that and dividing using binary fission, and waste will going to accumulate. Accumulation will increase with to the time because there is no fresh medium is being added. So this kind of setup will be there in the microbial growth curve when we see how much is the number of the viable cells with respect to the time. So in this there will be four phases. One is lag phase, exponential phase, stationary phase, and death phase. So we can see here in this picture that there are this number of bacterial cells in the log, and in x-axis with respect to time we'll see. So there are these different phases of the growth of bacteria. So in lag phase, which is the initial phase, so initially the bacteria cells will basically will not divide. It will try to sustain in that environment. So the lag phase is the initial the bacterial growth where the cells are basically they are metabolically active, but they're not going to multiply. So during this phase, the bacteria it synthesizes new enzymes and molecules to adapt to the growth medium, repair any cellular damage is there incurred before the inoculation, and it prepares for the DNA replication and cell division. In this, only the size of the microorganism will going to increase or will going to grow. So the size will increase, but it will not multiply. So statement 1 is correct here. In the exponential phase, which is the log phase, it is characterized by the rapid growth and logarithmic growth of the bacteria where the cell divides at a constant maximum rate leading to a constant doubling time. So it is exponential increase in numbers. Balanced growth means all cellular components used at consistent rate relative to each other. So here it will show constant doubling time, which is correct in exponential phase because at constant maximum rate bacteria will going to divide, and since here we are taking the binary fission case, so obviously the bacteria will double, so we can also say it as a doubling time. So the constant doubling time will be there. Then next is the stationary phase occur when the bacterial growth rate it equals the death rate, resulting in the plateau in the cell number. So the number of the uh, cells which are basically growing is equals to the number of the cells which are in under the death. So the plateau reason we can achieve in the stationary phase, which is due to the nutrition depletion, toxic waste buildup, or other factors like the competitive microorganisms will be there. So because of that, we can see stationary phase. So stationary phase occurs during depletion, which is also correct. Then death phase basically is the final stage where the death rate exceeds the growth rate. So many cell lose this viability and they'll die, and not all cell undergo immediate lysis. So here the basically the death will be more. So basically we can see the decline. So in lag phase there is the constant we can see where the only the size is increasing and numbers multiplying is not there, but in log phase exponential rise in the number we can see. Stationary phase where the death equals to growth, and decline phase where the cell numbers decreases. So here in the death phase, it involves immediate lysis of all cells, which is wrong because there is not immediate lysis of all the cells. So if we see this rate of the increase of cells is equals to constant, which is the rate constant multiplied by the number or mass of the cells. So here this is growth rate constant and is basically an index of the rate of growth. So basically this kind of microbial growth can be considered under first order autocatalytic chemical reaction. So it is similar to first order reaction. So basically this equation also predicts a straight line relationship between the log of the number of cells, which is n, with respect to time, if we see, and if we plot logarithm, so it can be in the straight line, it can form the straight line. And an ordinate intercept of with the ordinate intercept of log and naught. So it can be log, like if we take antilog, the equation can be n equals to n naught 10 to the power this growth rate constant multiply t minus t naught, which is initial to final time minus initial time, divided by 2.303. So mathematically we can write this equation like this, because we are saying that this microbial growth basically graph, this basically or growth rate is similar to a first order chemical reaction. So here only 1, 2 and 3 are correct and fourth option is not correct here. Okay, let's discuss this question, which has in column 1 food safety hazards like trans fats, heavy metals, bones and shells, and mycotoxins, and in column 2 we have the respective type like processing hazard, physical hazard, chemical hazard and biological hazards. So anyone who would like to answer this question? About that, so we have different hazards like mycotoxins, bones and shells, heavy metals and trans fats. We need to tell which one comes under hazards. Okay, let's discuss this solution. So here answer D is correct. So trans fat is basically a kind of processing hazard. Then heavy metal is basically a kind of chemical hazard. Bones and shells, it is a physical, and mycotoxin is a basically a biological hazard. Have different type of hazards as we saw. So microbial hazards are basically because microorganisms, if it can be under in the food like the bacteria, viruses, parasites or fungi, they can be basically can introduced into the food and can cause a food safety hazard, and it can be like these harmful bacteria can cause basically severe illness in the human body. So these are the microbial hazards. Next one is a chemical hazard. So chemical hazards basically includes some kind of the toxic chemicals which can come in the food and can cause threat to the human health. Then is the physical hazard. So physical hazard can include like the foreign object coming into the food, like hair, or here it's like shells like that. All these can come into the food. And processing hazard means that if like when we are starting with the food product, all the substances are safe to consume, but while processing, like at the high temperature, the fats can convert into the trans fat, and it can be of having negative health impacts. So it can come under like the hazards which are occurred due to the processing. So it is under processing hazard. And also we can have some kind of allergens like the proteins, or some kind of allergic allergens, or the some like basically proteins are there which basically going to produce or trigger some kind of allergies into some human individuals, so that they come under the allergens. So these all will going to or result into the food safety hazard. Like if we see the physical hazard, it can be like stem, seeds, hair, bone fragments, feathers, jewelries, nails, then any kind of bull like the nuts and the bolts, then nails can be there, or jute strings can be there, buttons can be there, cigarettes, matchsticks will can be there. So these kind of things, if they're in the food, it can be considered under physical hazard. And chemical hazards basically includes like the pesticide residues, then veterinary residues, non-permissible food additives. If we have given some food additives which are uh, beyond the safe limits, they can also be come under this chemical hazard. Then cleaning agents, the CIP cleaning if we are doing, and some kind of the cleaning agents can be there in the machine, and it can may come into the food product. Then adulterants can be there, and if some additives will be beyond the accessible or permissible limit. Then microbial hazards may include due to bacteria, yeast, protozoa, viruses, molds, parasites. So they can come under basically microbial hazards. So basically food safety hazard means that any factor which is present into the food and that has a potential to cause harm to the consumers, either by causing illness or injury. And hazards are those factors that the food safety seeks to protect against or contain or eliminate from the food product. Most of the unsaturated fatty acids in natural triglycerides are present in, but they are converted into their trans form when oils are basically heated at high temperature during refining or hydrogenation process. So these trans fats, they are not good for health. So that is the processing hazard. Then heavy metals, which includes lead, mercury, arsenic, and cadmium, they are the toxic elements that are contaminate the food through the soil, water, or the industrial pollution. So it may lead to heavy metals poisoning in the human being. So they can come under the chemical hazards. So lead and mercury in the seafoods, they cause the neurological problems. Arsenic is linked to the skin lesions and cardiovascular diseases and cancers like the skin, bladder, or lung cancer. Cadmium can affect the kidney dysfunction and can lead to bone demineralization. So it is also known as Itai disease. Then bones and shells, if they are coming into the food because they are remained in the processed meats or seafood products, So it can cause injury and dental damage and secondary infections, and they come under the physical hazards. And then mycotoxins, which are the toxins produced through the secondary metabolites produced by the molds, such as the aspergillus, penicillium, and fusarium. So these can come under the microbiological hazards. So common example, if we see, so aflatoxins are there, then ochratoxins and fumonisins are there. So this aflatoxins and ochratoxin A that can be produced from the aspergillus strain, and fumonisins they can be produced from the fusarium strain of the fusarium type of the bacteria. So they can cause, these are the toxin names. So these can cause, these mycotoxins, then they can cause nausea and the vomiting. And if there is severe intoxication is there, so they can damage some organs like kidney, liver, and it can also result in the hepatotoxicity and also nephrotoxicity and also the carcinogenicity. So they can basically cause mild to severe symptoms also. So here trans fat includes the processing hazards, heavy metals includes the chemical hazards, bones and shells they include the physical hazards, and mycotoxins they are the biological hazards. Okay, let's solve this question now, which is regarding the bacterial population and the doubling time. So calculate the bacterial population, so which is in CFU per gram after 5 hours, and the mean doubling time, which is given as 30 minutes. And the initial population, which is N naught, is given as 10 to the power 10 to the power plus 3 CFU per gram. So please solve this question and let me know answer. Anyone be solving this question? So Lakshmi has already solved this question, and as per her, answer is correct one, which is 1.024 into 10 to the power 6 CFU per gram hours. Anyone else who has solved this question? Okay, so basically she's correct that option A is basically correct here. So basically the generation time or the mean doubling time is of an organism or microorganism is basically the time it takes for a bacterial population to double in number and calculate and can be calculated during the exponential phase where the binary fission is happening. So this is a parent cell, and when the cell division will happen, two daughter cell will going to produce, because we are considering it as a binary fission. So G, mean doubling time or the generation time, is equals to the growing time T divided by N, which is number of generations. So to determine the generation time, microbiologists they plot the natural logarithm of cell number against the time using a semi-log graph, and where the slope is going to be equal to 0.301 per gram, and number of generations if we see is equals to 1 upon ln2 is equals into ln n by n naught, which is n is the final population of the cell and n naught is the initial population. So here one cell is basically dividing into 2 by binary fission. So that's why we are using this formula NT, which is um the population of the cell after some time is equals to n naught initial population into 2 to the power n. So here n is basically the number of generations, how much generations we are considering here. So let's come to this question. So in this question we need the bacterial population which is after 5 hours. That means we need to calculate n. So n basically is equals to as we see here n is equals to n naught into 2 to the power n. So n equals to n naught into 2 to the power n. So n naught is basically, which is the initial population, is 10 to the power 3 CFU per gram. So that is 10 to the power 3 into 2 to the power n. So here 2 into n, and we need to calculate how much is the generations. So here we don't know n. So n using the above formula, which is this, that is the generation time or the doubling time is equals to growing time divided by number of generations. So number of generations n is equals to growing time t divided by g, which is generation time. So t divided by generation time or doubling time. So n is equals to, which is basically the number of generations is equals to n is equals to t, which is the time for growing. So that is basically 5 hours. So 5 hours is 5 into 60, that is 300 minutes, which is divided by the doubling time, which is 30 minutes. So divide by 30 minutes. So this will be cancelled. This will be come as 10. So 10 is the number of basically generations. So here we can put 2 to the power n, which is 10, which is the generation time, the number of generations it will take. So if we see here, 10 to the power 3 is 1000 and 2 to the power 10 is 1024. When we multiply, we can get 1.024 into 10 to the power 6 CFU per gram, which is option number A. So it is here option number A is correct. Okay, so everyone is clear with that question, previous question. So can we move to this next question about d value calculation? So next is this d value calculation. Okay, so here it is saying, the question is saying, if a thermal process reduces the microbial count of a food from 10 to the power 6 CFU per gram to 10 to the power 3 CFU per gram in 15 minutes, calculate the d value in minutes. So here microbial population is coming 10 to the power 6 CFU per gram, to it is basically coming down to 10 to the power 3 and it is taking 15 minutes for this. So we need to calculate d value. Anyone who is calculating this question? I'm so sorry. So Lakshmi has already calculated and she's saying B is the correct answer. Okay, let's see. So A is the correct answer here. Okay, so let's discuss this question now. So basically, first we need to understand that what is microbial survival. Basically, microbial survival curve is having on y-axis the microbial population, on x how much time it is taking to reach that how much like time incubation time, what is a microbial population is there. So it basically follows usually first order equation. So that is first order equation we can write dn by dt equals to minus k n to the power n. So n small n is basically equals to 1 here because it is a first order equation. So it will be dn by dn by dt equals to minus k n. Then if we solve this, it will be n2 equals to n1 e to the power minus kt. So where k will be the rate constant and n is the order of this model equation. Basically, n is equals to 1 because order of this equation is we are taking as the first order reaction. So this general model basically describes the reduction in the microbial population, which is n, as a function of time. So this is microbial survival curve. So what is d value then? So when convert this curve in basically the log, so this will going to be follow the straight line if we convert it into the log. So the time, the d value decimal reduction time is the time required to destroy 90% of the organisms. So d value is numerically equal to the number of minutes required for the survival curve to traverse one log cycle. So this in microbial survival curve, how much time is taking to traverse one log cycle. So that is from this to this is a one log cycle. So to reach from this to this or from this to this population, how much time is taking, or we can say 90% microorganisms we are destroying. So how much time is taking? So it is basically, if we see here, it will be this to this. So to reach this population, how much is time is taking? So that is t equals to 0. And here around maybe t equals to t1 or 6. How this is the one log cycle is happening. So we can see this time which is to get this population, which is d value. So mathematically it is equals to the reciprocal of the slope of the survival curve and is a measure of the death rate of an organism. So d value equation is basically log n1 minus n2 equals to t by d. So which we can see here that to from this population to this population, so which is this, so to transfer this, so that is log n1 minus log n2 because this scale is in log scale equals to t by d, which is basically how much is the time taking to get this much, which is we can see slope of this equation will be y2 minus y1 divided by x2 minus x1. So y2 minus y1, that means this minus this, which is log n1 minus n2 divide by x2 minus x1, which is this time. So it is d. So d is equals to divide by log n1 minus log n2, and also we can write this equation as n2 by n1 equals to 10, which is we are taking anti-log, minus t by d. So if we are comparing equation 1 and 3 with this equation and this equation, we can see here that k will be equals to 2.303 by d, or d equals to, basically if we put in the equation, it will be d equals to 2.303 by k log n by n naught. And F value, which is the thermal death time, is basically the total time required to accomplish a state reduction or a stated reduction in the population of vegetative cells or spores. So that means F value is basically the time. If we are, I am doing this much of log reduction, like I am doing 4 log reduction, or if I'm stating some log reduction, that is the F value. And if I'm saying just to transverse one log cycle, that is D value. So this time can be expressed as a multiple of D values as long as the survival curve follows the first order model. So for example, a 99.99% reduction in microbial population would be equal to basically the 4 log cycle reduction, or F equals to 4D. So a typical thermal death time in thermal processing of stable shelf stable food is basically the F equals to 12D. So if we are saying some product is a shelf stable, or so F equals to 12D basically value, or 12 log cycle reduction, with the D value for Clostridium botulinum. So its formula is basically log n1 minus log n2 equals to F by D. So if we talk about this question, so in this question we have given, it is given basically the initial microbial count, which is 10 to the power 6, and final microbial count is 10 to the power 3, and the process time is 15 minutes to get this much of reduction. So F equals to D into 10 to the power, F equals to D into log n1 minus log n2. So basically D will be F divided by this log n1 minus log n2. So F value is given as 15. So 15 divided by log n1. N1 is the initial microbial 10 to the power 6, and n2 is the final microbial count, which is given in the question as 10 to the power 3. So log 10 to the power 6 minus log 10 to the power 3. So log 10 to the power 6 will be 6, and log 10 to the power 3 will be 3. So 15 divided by 6 minus 3. 6 minus 3 will be 3. So 15 divided by 3 is 5 minutes. So we get 5 minutes basically the D value time to to have this kind of system. Is this question clear to everyone? Can we move to next question? Okay, Lakshmi, yes. So it's not 6 here, it's 5. Others, are you guys following us or solving these questions? Okay, let's have this next question. If an increase in the process temperature from 100 to 110 degrees Celsius, so we are increasing T value here, that we are increasing the temperature, which is T1 to T2, we are increasing temperature. So we can say that if we are increasing the temperature, so D value or time to reduce the microbial population will decrease. for 1 log cycle. So it is decreasing from 5 minutes to 0.5 minutes. So we have D1, which is 5 minutes, and D2 is 0.5 minutes. So we need to calculate here the Z value. Anyone who would like to solve this question? This is the simple, we need to put in the formula and we can solve this question. Lakshmi A 10, Anjali also A 10. So they both are correct. Basically, Z value, thermal resistance constant, is basically the slope of the TDT curve, which is thermal death time curve. So how we get TDT curve? So in TDT curve, we have on y-axis is the D values, and on x-axis we have the temperature values. So basically Z value says to get 1 log cycle change in D value, how much temperature is taking, that is Z value. So the number of degrees, which is in degree Celsius or Fahrenheit, required to bring a 10-fold change in the decimal reduction time. So it is the basically Z value is basically the temperature value, which tells that for transversing 1 log cycle change in D values, how much temperature we need to change. So Z is equals to, basically if we see Z, which is the slope of this line. So slope is basically here. If we see, that is Z value is equals to T2 minus T1 upon log D1 minus log D2. So it is given that T1 is 100 degree, which is given in the question. T2, which is final time, is 110. So we are increasing the temperature here, and initial D value is 5 and final D value will be 0.5. Z value will be basically T2 minus T1 upon log D1 minus log T2. So T1 is basically 100. So sorry, T2 is basically 110. So 110 minus this value and minus 100, which is T1, divided by log D1 minus log D2, which is log D1 is log 5 minus log 0.5. So 110 minus 100 will be going to give 10, and log 5 value is 0.698, and log 0.5 value is minus 0.301. So minus and this minus will going to be plus. So this is plus 0.301. So 0.698 plus 0.301 will be 0.999. So 10 divided by 0.999 will be 10.01 degree Celsius. So we are considering around 10. So 10 degree is the Z value. 10 degrees Celsius is the Z value here. Is this question clear to everyone? We are moving to next question, which is also a numerical type question about canning process. So a canning process is there. So which is conducted at, basically canning is conducted for the sterilization. So we are conducting it at 121 degrees Celsius, and the thermal resistance, which is D value of the Clostridium botulinum spores at 121 degrees Celsius, is given as 0.21 minutes. The process needs to achieve 12 log reduction of C botulinum spores to ensure safety. So calculate the F value for this process. So here we need to calculate F value, and it is given that we need to achieve 12 log reduction. 12 log reduction. And D value is given already as 0.21 minutes. Temperature also given, that we are processing all these at 121 degree Celsius. Anyone who will tell me what is the F value here? Okay, we got two answer as C. So one name is I cannot figure out. So Anjali C. Which is 2.52 minutes. Okay, let's see the solution. So in canning process, what we do, we have some jars. We will going to put the sterilized pickles or some food products, and we'll have one like lid which will be put, and we'll like basically hermetically seal these lids, and basically after that we'll apply some temperature so that the whole container will going to be sterilized. So we can have this kind of packaging for canning. So in this question, we have this D value given for Clostridium botulinum spores at 121 degrees Celsius, which is 0.21 minutes, and log reduction is 12 log reduction. So we know that F is equals to D into log N by N naught. So F equals to D, which is T is 0.21, and the log reductions are already given, that we are going for 12 log reductions. So 0.21 into 12, which is 2.52 minutes is the answer C. Okay, question clear to everyone? Now let's solve this next question. If the Z value of the process is degree Celsius, Z value is given 10 degrees Celsius, and the D value at 100 degrees Celsius is given as 5 minutes. So we need to calculate the D value at 110 degree Celsius. Anyone is solving this question? Please solve this question. Okay, so I'm discussing the solution. So Z value is given as 10 degrees Celsius, and the initial temperature is 100 degrees Celsius. The final temperature is 110 degrees Celsius. Initial D value is 5 minutes, and we need to calculate the T value. We know that Z value formula, Z equals to T2 minus T1 upon log D1 minus log T2. So we can shift this side this, and here Z value we can put in the denominator. So log, it can be written as rearranged and can be written as log D1 minus log T2 equals to T2 minus T1 upon Z. Now I can take this D2 in the initial, so I am taking both, like multiplying both sides with minus. So log, it will be plus after this, minus minus plus, so log d2 in plus and log d1 in minus, minus log d1 equals to minus t2 by t1 upon z. Now this is in minus, so I can written it in the form of the divide. So log d2 by d1 equals to t2 minus T2 minus T1 minus T2 minus T1 by Z, which is similar here. Now I can take this log in the anti-log form, like this. So d2 by d1 equals to 10 to the power minus t2 minus t1 upon z. So this d2 will be here and d1 will go here and in the multiply. So d2 equals to d1 into 10 to the power minus dt2 minus t1 upon z. Then we know that d1 value, which is this value, is 5 minutes. So 5 into 10 to the power minus T2 minus T1. So T2 is 110 degree Celsius and T1 is 100 degree Celsius. So 110 minus 100 degree Celsius divided by Z. Z value is 10 degree Celsius. So here I can write as 10. So D2 will be 5 into 10 to the power minus. Now 110 minus 100 will be 10 divided by 10, which will be come as 1. So 5 into 10 to the power minus 1, which is 0.5 minutes. So here B answer is correct. Anyone has problem in this question, or this question is clear to everyone? Can we move forward to the next question? Okay, so next question is about food spoilage. So basically, food spoilage type in column 1. So we have ropiness in the breads. Like this is the ropiness in the breads. Then putrefaction of the meat. So we can see here is a putrefaction of the meat, and then ropiness of the milk and cream. So it is a ropiness in the milk and also in the cream. We can see rope kind of formation is there. Then soft rot in vegetables. So this is soft rot in cabbage, the vegetable. In column 2, we have the spoilage causing microorganisms. So we need to match here. Anyone who would like to answer this question? Let's see the solution. So here answer B is correct. So two type of spoilage, bacterial spoilage and the fungal spoilage. In the fungal spoilage, spoilages like due to mold and due to yeast. So the bacterial spoilage, they basically require water activity which is greater than point. So because bacteria grows at water activity level and a more neutral pH, which is 6.5 to 7, they need to proliferate. So it can be found particularly in the products like milk, meat, vegetables, and the fruits, this bacterial kind of spoilage. And if we talk about this first, which is ropiness in the bread, it is caused by Bacillus subtilis. So this organisms, it produce extracellular polysaccharides which create a slimy, sticky texture in the bread, giving a ropey kind of appearance. So second, so this is related to Bacillus subtilis. Then Proteus vulgaris is basically a facultative anaerobe and is common spoilage bacterium which is involved in the proteolysis or putrefaction of the meat, so which is leading to or resulting to the characteristic odor of the spoiled meat. So basically this putrefaction of the meat, it is related to Proteus vulgaris. Then ropiness in the milk and cream, it is associated with the alkaligens viscolatis. So it is basically result in the formation of slimy or ropey consistency in dairy products, especially due to improper refrigeration. And next is the, so this ropiness in the cream, it is associated with this B, and then soft rot is associated to D. So soft rot in vegetables is caused by Irvinia carotovora. So it is basically leading to the tissue maceration, making the vegetables soft and waterlogged. So in these two tables, we can see different food products and the spoilage and what are the microorganisms involved. So bread, if we see, there is ropiness, that's Bacillus subtilis, and the putrefaction of the meat can be caused by these kind of species. And cured meat, souring or greening or slimy can be formed from all these species. So if we see the ropiness in the cream or the milk, it can be caused by all these bacteria. And if we talk about in milk cream, if decomposition of fat will be there, so it is mostly by this kind of bacteria. And in milk, if there is more alkali formation, then this, and if off flavors will be there, then this bacteria. And in vegetables, soft rot will be due to the pseudomonas. So all these bacteria are involved in the spoilage of these kind of food products. Okay, let's discuss this question, which is the spoilage due to the fungus. So which statement is correct regarding that? Statement 1 says that the molds prefer lower water activity environments for their growth. Statement 2 is about yeast. So yeast thrives better in oxygen deprived conditions. Then Aspergillus species, they produce the mycotoxins, and Fusarium species, they spoil the pasteurized milk and high moisture dairy products. So we need to match. Okay, so which one is the correct answer here regarding the fungal spoilage? The second kind due to the fungal spoilage, which means due to the mold or the due to the yeast. Anju is saying C, which is 1 and 3 options or the statements are correct here, and 2 and 4 are the incorrect statements. Anyone who would suggest us any other option, or is this option is correct? Okay, we are discussing the solution part. So statement 1, 2, 3 are correct here because in the molds basically grow on the food surfaces because they need the free oxygen. Also, they grow in the broad range of pH from pH 2 to 8.5, but mostly in the acidic, and also in the low water activity level, which is zero point 0.7 to zero point 0.8. Mold commonly spoils basically the dried food products like grains, beans, peanuts, and pulses. So the molds basically thrive in the low water activity condition because they can extract from the environment and they can survive where the bacteria cannot perform these functions. So statement 1 is correct here. Then yeast, basically under anaerobic conditions, yeast ferment sugars to ethanol and CO2, which is carbon dioxide, making them to thrive at anaerobic conditions. They grow within a water activity range of 0.9 to 0.95, and they can tolerate a broad range of pH, which is 3 to 8, and mostly they grow in the preferred environment is acidic environment, which is 4.5 to 5.5. So yeast basically can grow in the anaerobic conditions. Statement 2 is correct here. Then aspergillus, basically species, they are basically the mold or fungi. They're known for their potential to produce harmful mycotoxins under the specific conditions. And in the aspergillus, these species, there are different mycotoxins they produce, like aflatoxin, ochratoxin A, then patulin, citrinin, then aflatrim, then it can produce like many, and this number is not just limited to this. This number can be for, like, this is just few examples of this. So this aspergillus species, it produce hundreds of the mycotoxins. So cyclonic acids, and then cyclopazonic acids, then pterins, then gliotoxin, then also this metoxins. So basically, these are like different kind of toxins which are produced by aspergillus species. Also, uh, because this aspergillus, they are basically going to form the spores also. So these spores, what will going to happen is, these spores, they will going to be into the like they will go in the respiratory tract or respiratory system in the human body, and they can cause infection. That infection is known as aspergillosis infection, which affects the human's respiratory system. So this aspergillus species, they produce mycotoxin, which is correct. Then fusarium, this is a fungi and it can grow at low water activity levels. But in milk, if we see, and the other dairy products, they are very highly in moisture content and moderate in acidity. So that fusarium species cannot grow in the dairy products. So basically the fusarium, they are found in the evidently present in the food microbial community, and also they exist as a commensal member of the skin microbiome. So basically in our skin, so the some fusariums can be present in the as a commensal member. That means, what is a commensal member? So it is basically, it will be on our skin and it will get some benefit out of from our skin, but it will not harm our skin. So when one partner is getting the benefits, the positive effects, but another partner will have no effects, then it will be in commensal member. So, and this fusarium species, some of the species can produce some toxins also. So that includes fumonisin, then trichothiocin family toxins, and zearalenone kind of toxins it produces. So basically, it cannot grow in high moisture dairy products. So statement fourth is correct. 1, 2, 3 statement are correct here. So if we see the diagram, so this is the basically Aspergillus species. So it looks like this. So it will have these spores, and if you are working with Aspergillus species, these spores can go into your respiratory tract and can cause severe infections. And this is the fusarium, how it looks under the microscope. So these are the different foods, and they are spoiled by the fungus. So different fungus. So basically, if you see, they are like different breads. So molds can form because of penicillium and rhizopus. So like these, there are different uh, in milk, basically milk or cream. Basically, yeast and mold can grow for their decoloration or fat. So like this. Let's calculate one like numerical question now. So calculate the water activity of a food with a vapor pressure of 15 mm Hg at 25 degrees Celsius. Given is the vapor pressure of pure water is 20 mm Hg at the same temperature. So we need to calculate here water activity. Anyone who is solving this question? Okay, let's solve this question. So water activity basically is the ratio between the vapor pressure of the food itself when in the complete undisturbed balance with the surrounding air medium and the vapor pressure to the distilled water under the identical condition, which is P by P naught. So vapor pressure of the food is given as 15 mmHg. So 15 divided by the vapor pressure of the pure water, which is 20 mmHg. So 15 divided by 20 will be 0.75. So B answer is correct here. And because of the different water activity level, different kind of bacteria or the molds or the yeast can grow. So bacteria can grow greater than 0.91 water activity level, yeast after 0.88, and molds 0.7 to 0.88. I hope this question is clear to everyone. So which of the following statement is correct with respect to the oxygen dependency of microorganisms? So first statement is oxygen act as a antimicrobial agent for the obligate anaerobes. B is obligate anaerobes can survive in absence of oxygen. Third is aerotolerant anaerobes remains unaffected by the change in the oxygen level. And fourth is facultative anaerobes grow better in presence of oxygen but can live without even without it. So we need to tell what is correct here. Anju, C is for this question. 1, 3, and 4 are correct. So the microorganisms basically are grouped based on their. So she is correct. 1, 2, 3 are the correct answer. So basically, microorganisms, they are grouped based on the oxygen requirements. They can be aerobes, anaerobes, or facultative anaerobes. So basically, based on how much oxygen they need for their growth, a microorganism can be divided into the different classes. So the obligate aerobes means that they need oxygen. So they are dependent on aerobic respiration for making their energy. So they cannot survive without oxygen. So they must need oxygen for their growth. So this is basically aerobes. So they grow here in the oxygen environment at the top. So if we see, if we are having some also, because here is the some oxygen in the upper atmosphere, so oxygen concentration will be high at the top and will be low at the bottom. So obligate aerobes will be at the, they'll grow at the top surface. So facultative anaerobes are versatile microorganisms that grow in both the presence and absence of oxygen. And in the presence of oxygen, they basically use the aerobic pathway for their respiration, which is more efficient for ATP production. So oxygen, so they'll switch to anaerobic respiration or the fermentation pathway to sustain themselves with the low energy yield. So they are basically, they can grow in the whole, like from the top to bottom, they can grow. So if they'll get oxygen, they can grow. If they'll not get oxygen, they can grow then also. Aerotolerant anaerobes, they do not use oxygen for their metabolism but can survive and grow in its presence. So, aerotolerant anaerobes, they are basically don't like oxygen, but when there is oxygen, they can still grow. So, they perform mostly the fermentation regardless of the oxygen level. Their tolerance is due to the presence of enzyme like superoxide, dismutase, and catalase, and which detoxifies reactive oxygen species. So they are basically are the one which do not switch their uh, ATP production pathway. They are using fermentation only for their ATP production, but they have these kind of enzymes which can detoxify the oxygen. So they can grow in both the condition, but they don't like oxygen. So they can grow in both this. And obligate anaerobes, they cannot survive in the presence of oxygen. So they need compulsory anaerobic conditions because they lack the necessary enzymes also like the superoxidase and catalase and to neutralize these toxic reactive oxygen species like the superoxides radicals. So exposure to oxygen can lead to the oxidative stress and damaging the cellular structures and killing the microorganisms. So they will grow at the bottom only. And there is one more which is microphilic aerophilics. So they need less oxygen for their survival. So they can grow in between of the medium. So here, first statement is saying that the oxygen acts as an antimicrobial agent for the obligate anaerobes. So obligate anaerobes, they cannot survive in the oxygen. So this statement is correct. Then obligate anaerobes, they need basically oxygen to for their survival. So correct. And then anaerobes, they remain unaffected by the change in the oxygen level. So which is also correct. And fact and anaerobes, they grow better in the presence of oxygen. Still, if there is no oxygen, they can survive based on the fermentative pathway, which is also correct. Which of the following statement is a correct here regarding the foodborne infection and food intoxication? So foodborne infection is the result of the consumption of food containing toxic substances, and presence of harmful microorganism in the food is a major reason for food intoxication. So we need to tell which statement is incorrect here, not correct here. Anyone who would like to answer this question? Anju is saying B, only two. Okay, let's see. So answer is here, neither one nor two. So both statement are not incorrect. So they are basically the correct statements. So foodborne illnesses are basically the acute illnesses which is resulting from the intake of the contaminated food which is having the harmful pathogens or either the toxins. So foodborne infection, it causes primarily due to the ingestion of the live microorganisms like the bacteria, viruses, fungus or the parasites that can go and multiply in the host cell, which gives the problem or the health complications in the host cell. Like the salmonellosis, listeriosis, E. coli are the common microorganisms that are responsible for the food infection. So this can also transmit from one person to another, and it can have the symptoms like nausea, vomiting, diarrhea, abdominal pain, fever, and the dehydration. Whereas in if we see this is the food infection. So it can be because of bacteria, virus, and the parasites. And foodborne diseases, it is because of the foodborne intoxication. So food intoxication is basically due to ingestion of the toxins which are produced from these microorganisms due to the microorganisms itself. So because of the toxins or the toxic compounds it produces. So botulism, staphylococcal food poisoning or aflatoxicosis are the primary examples of the food intoxication, and it is non-transferable, and its symptoms may include nausea, vomiting, diarrhea, weakness, respiratory failure, numbness, sensory or motor dysfunctions. Also, the food infections, these food-borne infections, they are classified as a bacterial, mycotic, or viral. So it can be based on the viruses if it's there, like hepatitis A, or it because of the bacteria like cholera and all, and based because of the mycotic means the because of the fungal sources. So it can be because of the candida species and all. So here both statements are correct. So because of the food consumption of the food which is having the toxic compounds in them, and also food intoxication is because of microorganisms in the food which are producing the toxins. So it can have food intoxication can because of the chemical compound contaminations like heavy metals and all, and natural toxins which are in the mushrooms like and bacterial toxins and fungal toxins. So which of the following describes effective waste management in food facilities here? So we have four stages, that segregation of organic and recyclable waste, then disposal only when bins are full, and storing waste near food processing area, and fourth is regular disinfection of the waste bins. Anyone who would like to answer this question? Uh, is all the questions clear so far, or you have any doubt in the questions which we haven't discussed? Okay, so in this, basically, effective waste management in food facilities, it can be basically done by segregation of organic and recyclable waste and regular disinfection of the waste bins, which is first and fourth. So effective waste management in food facilities involves the segregation of organic and recyclable waste to ensure proper disposal and promote recycling, as well as regular disinfection of the waste bins to prevent the microbial growth and the contamination. Whereas the waste should never be stored near the food processing area, and because to avoid the cross contamination, also it should be disposed of promptly even before the bins are full, because of the maintenance of hygiene and prevent odor. So statement first and fourth are correct here. So if we see the waste management protocol, so waste segregation, so that means segregation of the waste like organic, recyclable, journal waste, and store them in appropriate or the clearly labeled bins. We should segregate these waste, and we should regular dispose before the overflow and the potential contamination will cause to food. So before the overflow, we should properly regularly dispose. And bin also should be cleaned and disinfected regularly so that the bacteria and the odors will not grow. Okay, next is the question related to HACCP. Statements are correct. So statement 1, 2, 3, and 4 has the HACCP ensures food safety by identifying the potential hazards. HACCP focuses exclusive on microbial contamination. Third is it includes monitoring and corrective actions for CCPs, critical control points, and the HACCP is a reactive, non-preventive approach. Anyone who would like to answer this question? Okay, so we are discussing the solution of this question, which says that first and third is the correct answer. So basically, HACCP in this H stand for hazard, A for analysis, C for critical, then C control points. So hazard means that anything which is causing health hazard, and A means we need to analyze the hazard and then investigate about the hazard. Then critical means the crucial, which is for containment, and then control, we should handle those points of the critical points to analyze and to reduce the hazards in the food product. So HACCP is basically a systematic and science-based system designed to ensure the food safety by identifying, detection, and controlling the potential hazards that could affect the food safety at critical points in the production process. So basically, statement 1 is correct here, that it ensures the food safety by identifying the potential hazards. It aims to prevent, eliminate, or reduce the food safety hazards to the acceptable limits. And the CCPs, they basically are for the implementing the effective monitoring and corrective actions. And the HACCP basically ensure the production of safe food, protecting the customer health and complying with the regulatory requirements. It's, so basically, it includes monitor corrective actions for critical control points. And it is basically a preventive method. So statement 4 is incorrect. And it basically focuses on all kind of hazards or the contamination, just not the microbial hazard. So statement 2 also incorrect. And 1 and 3 are correct here. So HACCP, they are having the 7 principles basically. And 5 basically are the preliminary steps. So it includes 5 preliminary steps, which means that assembling the HACCP team, then describe about the product, then identify intended use, and then fourth, construct flow diagram, and 5, verify the flow diagram on site. And after that, 7 principles, which means first is the conduction of the hazard analysis, and then second is the determination of CCP, third is establishment of the critical limits of these CCPs, and fourth is regular monitoring. So establish the monitoring procedure or the system for each CCP. And fifth is establishment of the corrective actions. establish the verification processes, or seventh is the establish of the record keeping and the document keeping processes. I hope this question is clear that first and three are correct here. Okay, now let's discuss about the probiotics. Which of the following statement about probiotics is correct? So probiotics enhance gut barrier properties by modulating immune responses. Then second is the probiotic are effectively consumed in the form of the fermented dairy products. Third is bifidobacterium species are commonly probiotic for digestive health. And fourth is probiotics produce antimicrobial substances that inhibit pathogens. So we need to tell which statement is correct here. Who would like to answer this question? Anju is saying D, which is all of the statements are correct. Anju, you are correct. All the statements are correct here. So probiotics basically, or the beneficial microorganisms, they basically support the gut health by aiding digestion, producing vitamins, and preventing the harmful pathogens. They support the gut health through inhalation, pathogen inhibition, and reduce inflammation, gut microbiota balance. So they are basically modulating the immune response, and basically they inhibit the pathogens also. Fermented dairy products are a wide recognizable and effective delivery system for probiotics. So basically dairy products are usually fermented and usually probiotics are delivered in this, like the lactic acid bacteria, lactobacillus, lactococcus, leuconostoc, pediococcus, streptococcus. They can basically be used to ferment the dairy products. So basically dairy products can be used for probiotics, delivery of probiotics. So we can see here, we have the different classes of the bacteria, like the lacto streptococcus, bifidobacterium. So these are the strains which are basically the probiotic strain. So they basically provide targeted benefits like the antimicrobial activity and the digestive support to the human. So the microorganisms basically, so this statement is also correct that bifidobacterium species are common probiotics for digestive health also. So in regards to that, the probiotics are basically given in the fermented dairy products, because what happens, the microorganisms there can get sugar in the form of lactose and it will convert it into lactic acid. So the milk proteins can be coagulated because of the low pH, and because of that thick texture can be seen in yogurt and cheese. So this lactic acid lowers the product pH, limiting the other harmful bacteria or the microorganisms to grow, and thereby increasing the shelf life and the safety. Like if we see Lactobacillus bulgaricus, so it ferments milk to yogurt, and Streptococcus thermophilus, it works with the Lactobacillus and it will produce yogurt and the cheese, and Lactobacillus casei, it basically used to ferment the cheese and the fermented milk products. So if we talk about the probiotic and their gut microbiota modulation, so what happens? These probiotics, they will help to improve the gut microbiota because they help in nutrients metabolism, modulate host energy balance, then regulate inflammatory immune responses and enhance intestinal barrier functions and gut brain axis communication. So these probiotics, they balance basically the gut microbiota, enhance the digestion and immune function, and defend from the pathogens. And they are found in the fermented foods and the supplements, and they promote health by supporting the beneficial bacteria and inhibiting the harmful microorganisms. So the gut microbiome exert profound health effects on the health and disease status of the host, encompassing functional functions related to nutrition metabolism, host immune regulation, and potential impact on the central nervous system. Like if we see here, so in the from the if we see the probiotic action mechanism. So first is the like what is uh, nutrient will be there in the gut. So there will be the nutrients and the prebiotics, and the bioconversions will be there. After that, the production of growth substrates will be there, like the vitamins, and then direct antagonism effect is there. Then competitive exclusion will be there. Then barrier functions. After that, reduction of the inflammation and the immune stimulation at the end. So this basically this like this the these probiotics they function. So in usually probiotics need some kind of the prebiotics for their or the some kind of nutrients for their growth. So they will compete for this and their that they'll convert these prebiotics or the nutrients into some useful their bioconversions will be there. Then they'll produce some vitamins and all from these, and then they will have some like they'll fight with the harmful bacteria or the microorganisms. They'll fight so that they'll get the nutrients, and that's why they remove the harmful bacteria, and then they'll help in the further in the reducing the inflammation and helping in the immune system stimulation. Probiotics, they support the gut health. They by basically first the enhancement of the epithelial barrier, and by increasing basically the adhesion to the intestinal mucosa and inhibiting the pathogen adhesion, and then basically excluding the pathogens or the pathogenic microorganisms by the competition exclusion. Then it produces the anti-organism microbial or antimicrobial compounds, which basically remove these harmful bacteria or reduce their population, and then at the end they'll modulate the immune system. So they have this immune response. So these probiotics or the beneficial bacteria, they compete with the pathogen for the colonization sites and the nutrition. So they produce the short chain fatty acids compounds and the bacteriocins to inhibit the pathogen growth and support the immune system by promoting IgA production and regulatory T cells that releases the anti-inflammatory cytokines. Then these prebiotics, which are the non-digestible food components, they are basically fermented by these beneficial gut bacteria to produce the short chain fatty acids, and they provide the energy for the gut cells and enhance the gut barrier integrity. These prebiotics and probiotics, they're gonna protect against the pathogen and improve the gut barrier function and balances the immune response to prevent the inflammation. Then these probiotics, they also enhance the digestive health by maintaining a balanced gut microbiota, aiding in nutrition, digestion, and absorption. And it prevents the diarrhea by restoring bacterial uh, balance. And these probiotics, they also strengthen the gut barrier and preventing the pathogens and the toxins from entering the bloodstream, and they modulate the immune response to boost defense against infection and reduce the inflammation. Also, like they reduce inflammation because probiotics, they introduce the beneficial bacteria that out competes with the harmful bacteria, which produce also the anti-inflammatory compounds. The prebiotics, they provide nourishment for the beneficial gut bacteria, and they promote the healthy microbiome. So overall, if we see, so these probiotics, they are going to help the grow the intestinal. So these prebiotics help to grow the intestinal microbiota and overall to achieve the homeostasis of the gut microbiota and maintain the basically gut barrier integrity and modulate the immune response. So if we see here, so they're going to this probiotics, they're going to compete for colonization sites and the nutrients with the pathogen. So they'll produce some kind of bacteriocin and short chain fatty acids. So they'll not able to grow the pathogens. Then they'll modulate the immune response from the beta cells, or they'll produce cytokines, and they'll produce IgA like this. And then they also improve the gut barrier integrity by production of the mucin glycoproteins, and they provide the energy also by producing the energy giving compounds. So like this, they'll help to overall get the immune response and the gut microbiota homeostasis. Okay, let's discuss this question. We have in column 1 food products like sauerkrauts, bread, sausages, and yogurts. And in column 2, we have the beneficial kind of microorganisms which is used for the preparation. So we need to match here. So this is the sauerkraut, this is bread, this is sausages, and this is yogurt. They all are the fermented products. So Lakshmi is saying B here. So Lakshmi is correct. So basically, the Lactobacillus plantarum is basically responsible for fermenting the sauerkrauts, kimchi, pickles, or the vegetables, and they'll produce the lactobacillus. They produce the lactic acid, which basically preserves the product and it enhances its tangy flavor. Leuconistic uh, mesenteroids, they basically initiate the fermentation process in the sauerkrauts and the pickles, but usually it is done by Lactobacillus plantarum. And Saccharomyces cerevisiae, it is also known as the baker's yeast, which is used in the bread making, and it is used to ferment the sugars, and it will produce the carbon dioxide, which will act as a leavening agent, and because of that, bread will be fluffy or airy. Then Lactobacillus sakei is basically, it is playing a role in the sausage fermentation because it produces lactic acid and it will reduce the pH to enhance the flavor, and it inhibits the spoilage causing microorganisms also. Pediococcus acidilactic basically also ferments meat and produce lactic acid. And the last one is Streptococcus thermophilus. It is basically a primary culture in the yogurt production, and it ferments basically the lactose into the lactic acid, and it thickens the milk and give yogurt its characteristic tangy flavor. So this one is the Lactobacillus plantarum. Then this is the Saccharomyces cerevisiae, and this is Lactobacillus sakei, and this is Streptococcus thermophilus. So here answer B is correct. That sauerkraut is related to Lactobacillus plantarum. Bread is related to Saccharomyces cerevisiae. Sausages, Lactobacillus cepi, and yogurts basically Streptococcus thermophilus. So which of the following are examples of beneficial molds for food processing application? So here we have some molds, so we need to tell which molds are the beneficial molds and which are the spoilage causing molds. Anyone who would like to answer this question? Okay, so let's discuss the solution. Basically, 1 and 2, which is Penicillium roqueforti and the Aspergillus niger, they are the beneficial molds, and these two, Clostridium botulinum and Rhizopus stolonifera, is the spoilage causing. So, for the first one, which is Penicillium roqueforti, basically it looks like this. So it is used in the blue cheese, which is this is the blue cheese. So it is used in the blue cheese formation, like the Roquefort. So Roquefort is kind of a moist French cheese made from the sheep's milk, or it can be used for the formation of Gorgonzola, which is the Italian blue cheese, which is made from the unskimmed cow's milk. And the another is the Stilton, which is also a English blue cheese, which is made from the cow's milk. So it is used in these formation, this Penicillium roqueforti, and it imparts the characteristic blue-green marbling and sharp, tangy flavor. So, and the second one is Aspergillus niger, which looks like this. So it is used in the industrial production of the citric acid, which is widely employed as a food additive and a preservative. So this is also a beneficial use. The Clostridium botulinum, which looks like this. This is the Clostridium botulinum. So this is a harmful bacteria, and also the Rhizopus stolonifera, which is look like this. So it causes a spoilage of the bread. So these two are the spoilage causing microorganisms. And if we see the process diagram of citric acid fermentation process from the Aspergillus niger, so we take Aspergillus niger OE55. So it is basically being cultured and sporulated on the PDA slants at 30 degrees Celsius for 4 days. The spore inoculation will do uh, in 0.9% NaCl. After that, the fermentation is going to truffle shake flask, and the beet molasses is given as the substrate, and at 30 degrees Celsius for 8 days and 200 RPM. So this is the beet molasses and the other ingredients supplied in the fermentation medium, and then the filtration will be there for the supernatant. So as a supernatant, we get the citric acid and sugar, and at the residue, we'll get the biomass. So this supernatant will include the citric acid. So like this, the Aspergillus niger is used to produce the citric acid. So 1 and 2 are the beneficial molds. So next is which of the following statement correctly describes algae's role in the food industry? Which is first is carginin is used as a stabilizer in dairy products. Second is algae provides a low protein but highly sustainable food sources. Third is spirulina and cholera are common dietary supplements, and D is fourth is algae derived bioactives are used in the functional beverages. Anyone who would like to share his or her opinion on this question? Okay, let's discuss the solution. So basically 1, 3 and 4 are the correct answer here. So algae basically is a rich source of high quality protein and bioactives, and make that's why it is used in the diets as a supplement. So they contain basically essential amino acids, vitamins, minerals and antioxidants, and provide numerous health benefits. Algae's bioactive compounds offers anti-inflammatory, anti-cancerous and immune boosting properties. So algae basically plays a crucial role in the food industry as a source of carrageenan for stabilizing the dairy products and protein dietary supplements like spirulina and the cholera, and it can also be used as a bioactive for functional beverages. So algae is not a low protein source, so it is basically usually high protein, and exceptionally some varieties can be having the low protein, but most of the variety will not have the low protein. Basically, carrageenan is used as a stabilizer in dairy product, which is correct, and basically, sorry, this is incorrect. Second one is incorrect, and third one is spirulina and cholera, basically used as which is correct, and algae is used as a bioactive in the functional dairy, which is correct. So second statement is incorrect here, rest are correct. So basically the algae can be of like the different types: red algae, green algae and the brown algae. Spirulina is basically the high protein content algae, which is rich in vitamins and minerals. Cholera, they have all the essential amino acid, antioxidants and omega-3 fatty acids, and it is rich in fibers, vitamins and minerals. So these algae, they are basically rich in proteins, vitamins and other bioactive compounds like the fibers, omega-3 fatty acids. So they can be used as a good supplement for the food. So basically if we see the nutrient, so basically they are high protein content. So algae can contain up to 70% of the protein of the dry weight and have all the essential amino acids and vitamins. They have the rich source of vitamin A, C, E, B12 and minerals like iron, calcium and magnesium. So these are basically having omega-3 and omega-6 also, which are very good for heart and the brain health, and they also have the good antioxidant properties, so they combat with the antioxidative stress and reduce the inflammation. Then it also have the fiber, so it supports immune function and the gut health. Then they have the anti-inflammatory and detoxifying compounds also like chlorophylls and phycocyanin. So that is active compounds. Bioactive compounds are also there, so they can be used in different applications in the food industry like spirulina and cholera. They can be as a good dietary supplement of the high protein, essential amino acids, vitamins and minerals, and they can use as functional foods, so they can enhance the nutritional value of the food products. They can used in the aquaculture and animal feed, so that the fish which are fed with all these algae, they can be good nutrition and good health. So they can also be used as a sustainable protein source, which are different from the animal based proteins. And the food ingredients like agar and carrageenan and alginate, they can be used as thickeners, stabilizers and gelling agents in the food products. So today we discussed the topics like food microorganisms, like we discussed about bacteria, yeast and molds, and we discussed about their structure and metabolism, and then their reproduction and the food spoilage and poisoning microorganisms and food safety hazards. And we also discussed about the microbial growth, like the sources of microbial in the food and their microbial growth curve, microbial survivor curve, D value, Z value, F value. And then we discussed about the types of spoilage like the bacterial and the fungal. and how they can affect the human health, and the different factors like pH, temperature, moisture, and oxygen availability, these factors on the microbial growth, their impact on microbial growth. Then food poisoning and food intoxication we discussed, and about the HACCP and what are the different preventive strategies to have the good or the good hygienic, we can produce these food in the good hygienic food industries, or we can follow the good manufacturing practices or the good hygienic practices or the HACCP principles and the protocols to get the good food which is away from the food spoilage or the from the toxic microorganisms. And we discussed about the role of the microorganism as a positive way, which is the beneficial way, like they can use the fermentation of the breads and sauerkrauts and other things. And these probiotics, they can also use for the gut microbiota modulation and help to the reduce the inflammation. And they can the algae also can be, we have seen the algae can also be used as a good source of the proteins or the other bioactive compounds. In the next class we'll be discussing about chemical food additives, then functional food additives, food adulterations, toxics and the allergens, and international regulations on food additives. I want to discuss one thing at the end. Yes, so I want to discuss this question. So here, food infection is basically, we are discussing that food infection is the ingestion of the live microorganisms. So it can have food containing toxic compounds, but usually we have the microorganisms. So this statement is incorrect, right? Because that is, we should have the live organisms here. And in here, food intoxications, we have basically the toxins compound should be there, not the food, not the microorganisms. But we can have microorganisms also, like we can have the toxins produced by the microorganisms, and basically the microorganisms, mainly the toxic compounds. So here the harmful microorganism is given. So what if we see, the both statements are basically incorrect here. So here it is incorrect written. So which option are correct? That is neither 1 and 2. Both options are incorrect because food intoxications have the compounds like the harmful compounds, harmful toxic compounds in the food. Whereas foodborne infection means that food is containing with the not the toxic compound, but the itself the live microorganisms. So if we see like the proper statement. So please, when you are solving, remind this, keep this in your brain. Okay, so the recordings will be provided on this basically, and the PPT also will be shared in the Google Drive link, on the Google Drive link. So thank you so much for joining today's session. So this is the end of the session. So you can leave the meeting. If you have any doubt, please stay and ask your questions. Otherwise, you can leave the meeting. Thank you so much for joining today's class. And if you have anything to discuss, feel free to discuss. I want to see the attendance for today. Do you have anything to discuss or any questions or doubts? Please stay. Otherwise, you can leave. Thank you so much for joining today's class.